Good morning, children. Yesterday I have told you about the transforming principle. We were discussing about the transforming principle as such, and I have told you that the experiment was performed by Frederick Griffith on Streptococcus pneumoniae. There were two strains R and S, and finally in the end I was discussing that if the heat killed S strain is kept along with the live R strain, it is observed that the mice, instead of the result should have been mice should have been surviving, but the result was found that mice is dead. Now what was the condition why the mice was dead? Now Frederick Griffith assumed that there is some material which is still present in the heat killed S strain and which was able to convert the heat killed S strain into which was able to convert the live R strain into live S strain. That means the material which was stable enough and it was present in the heat killed S type of bacteria, it moved out and it entered the live R stain bacteria, converting the live R stain bacteria into the live S stain bacteria, resulting in the death of the mice. So there was certainly some substance, and that substance was the given name as transforming substance, and the principle was given was transforming principle. That means the substance which was responsible for this was transforming substance as such. And now after some time, biocharacterization of the transforming principle, the substance which was responsible for such a change, many scientists tried to prove that that particular substance is what kind of substance it is. It is DNA, it is RNA, or it is protein. So many scientists, they tried to prove whether it is DNA, RNA or protein, and finally the result came that it was DNA actually. But this result was not accepted by scientific community at that time. So naturally search for genetic material as a DNA continued. It was in 1952 that another scientist, Hershey and Chase experiment was performed by Alfred Hershey and Martha Chase in 1952, and they performed the experiment in bacteriophages virus. Bacteriophages viruses. Now bacteriophage virus, they can enter inside the bacterium cell as such, and this virus, the genome of this virus can enter the bacterium, and this genome used to get integrated with the genome of the bacteria. वायरस जीनोम जो है अंदर आने के बाद वो इंटीग्रेट कर जाता है बैक्टीरियम जीनोम के साथ, एंड आफ्टर दैट इट स्टार्ट्स टू सिंथेसाइज the protein particles and a genetic material, that is packaging of viruses starts to take place inside the body of the bacterium. So you can say that the genome of the bacteria, the genome of the virus, after getting inside the bacterium cell, it starts to forming its own copies. So with the help of this kind of virus, it was finally proved that DNA is the genetic material. Now we are going to see what kind of experiment was performed by Alfred Hershey and Martha Chase. ALF, name of the scientist was Hershey and Martha Chase. So now let us talk about their experiment which was performed. Let us talk about their experiment. Should I rub this? Okay. Now they cultured two bacteriophage in two different medium. They cultured bacteriophages in two different medium. In one medium, they provided bacteriophages with radioactive sulfur. Radioactive sulfur S35. Because in the medium sulfur was present, radioactive sulfur was present, but in the medium phosphorus was not radioactive. तो पहले केस में बैक्टीरियोफेज को जहाँ पे कल्चर किया गया, जिस न्यूट्रिएंट मीडियम में कल्चर किया गया, देयर रेडियोएक्टिव सल्फर वाज प्रेजेंट बट रेडियोएक्टिव फॉस्फोरस वाज नॉट प्रेजेंट. Similarly, in another medium, bacteriophage was supplied with radioactive phosphorus, that is the nutrient medium in which the bacteriophages were kept, that had radioactive phosphorus, but radioactive sulfur was not present. Sulfur was present but it was not radioactive in the second case. In the first case, radioactive sulfur was present, though phosphorus was present, but it was not radioactive. In the second case, just the reverse happened. Just the reverse was the constituent of the nutrient medium. Though radioactive phosphorus was present, radioactive sulfur was not present. Sulfur was present but radioactive sulfur was not present in the second medium. So bacteriophage was allowed to grow in this medium and it had radioactive phosphorus. Now this experiment has three stages. One is called as infection, in which the bacteriophage is allowed to infect the Escherichia coli. Now this is the cell of the E. coli or Escherichia coli. So this stage is called as infection in both the medium as such. S35 and here, similarly here also. This was done. First was infection, then followed by blending. First step was infection, second step was blending in both the cases. Blending means mechanical shearing in order to separate the bacteria with the infected virus. See, infected virus is just here. Now what happens during the time of infection? This DNA which is present inside the bacteriophages, this moves here as such and enters the E. coli cell. So it is something like this. Here you know that phosphorus is a part of DNA, important component of DNA. So naturally radioactive phosphorus will also enter if the DNA is entering inside it. And you see here all the phage particle is left outside and DNA has entered here also and here also. Here the DNA is radioactive in the second case, and all the protein particles, because you know the virus consists of only two things, protein subunit and DNA or the genetic material. So genetic material has entered the E. coli cell in both the cases, and the outer thing is because all the things cannot enter, the protein part cannot enter, it is left behind. And then the second step is blending in both the cases, in which the virus, which is now where the genetic material has already entered the E. coli cell in both the cases, only the virus protein coat is left behind. It gets separated. Separated मतलब ये देखो ये टच कर रहा है, यहाँ से separate हो जाता है, separate from the surface of the E. coli as such in both the cases. ब्लेंडिंग को इसीलिए हम लोग करते हैं, मैकेनिकल ब्लेंडिंग से इन दोनों को क्या करते हैं, सेपरेट कर देते हैं. और सेपरेट कर देंगे तो बट नेचुरल है, ये अलग अलग हो गया. अब देखो ये सरफेस पे नहीं रह गया है, ये अब सरफेस पे नहीं रह गया है, दैट इज ब्लेंडिंग. एंड देन वी परफॉर्म सेंट्रीफ्यूगेशन इन बोथ द केसेस. सेंट्रीफ्यूगेशन इज परफॉर्म्ड हियर ऑल्सो एंड हियर ऑल्सो. Centrifugation is performed at both the points. Both the cases centrifugation is performed. अब देखिए centrifugation perform होगा तो according to density, both virus which now do not have any kind of genetic material as well as the DNA as well as the bacteria will separate according to the density. And we know that density of the bacterium is more as compared to density of the phages. So naturally in the test tube, if that is done, you see the bacteria will settle at the bottom part in both the cases, and viruses will remain at the upper part. Let me tell you, this upper part is called as supernatant. इसको हम क्या बोलते हैं, अपर पार्ट को सुपरनेटेंट, और नीचे वाले पार्ट को बोलते हैं पेलेट. तो यहां भी सुपरनेटेंट बोलेंगे और यहां भी क्या बोलेंगे, पेलेट. दोनों ही केसेस में पेलेट, सुपरनेटेंट और पेलेट. तो यू सी in this case what will happen, the bacteria will settle down at the pellet region and in the supernatant region remains the bacteriophages. Now since S radioactive sulfur was added here in this medium, and radioactive sulfur is an important component of protein, it is not an important component of DNA. It is sulfur is an important component of protein, whether radioactive or not radioactive, but here in the medium we have kept sulfur as radioactive, so naturally it will become an important component of protein coat of the virus. And you know protein coat of the virus is here because only the genome has entered inside the bacteria, and genome here will not be radioactive since we have not used radioactive phosphorus in this case. We have used radioactive phosphorus in this case but not here. So naturally here, since we have added radioactive sulfur, and sulfur is an important component of the protein code as you can see here, naturally radioactivity will be emitted from the supernatant region and not from the pellet region. And this also proves that since sulfur is a part of protein, therefore protein has not entered the E. coli cell, proving that it is not the protein which is acting as a genetic material. अब क्योंकि यहाँ पे हम लोगों ने रेडियोएक्टिव सल्फर लिया था और सल्फर जो है प्रोटीन का पार्ट होता है, और हमने ये भी देख रहे हैं कि वो वायरसेस के पास ही रह गया है, अंदर वो एंटर नहीं किया है ई कोलाई के, क्योंकि रेडियो एक्टिविटी यहाँ पे कहाँ से इमिट हो रही है, सुपरनेटेंट रीजन से, और बैक्टीरिया कहाँ पे प्रेजेंट है, पेलेट रीजन से. इससे ये भी पता चलता है कि प्रोटीन जो है वो बैक्टीरिया के अंदर एंटर नहीं किया है और वो जेनेटिक मटेरियल नहीं है. लेकिन यहाँ देखिए, यहाँ पे हम लोगों ने रेडियो एक्टिव फॉस्फोरस लिया था, सल्फर प्रेजेंट था मीडियम में बट इट वॉज नॉट रेडियो एक्टिव. तो ये लेने का फ़ायदा ये हुआ, हमें ये पता है कि जो फॉस्फोरस होता है इट इज़ एन इम्पॉर्टेंट कंपोनेंट ऑफ डीएनए। डीएनए का इम्पॉर्टेंट कंपोनेंट है। आप जानते हो 2 न्यूक्लियोटाइड्स के बीच में फॉस्फोरस आता है, फॉस्फेट ग्रुप। तो अगर उसको हम रेडियो एक्टिव मान लेंगे तो नेचुरली जो बैक्टीरियोफेज अपने मीडियम से लेगा फॉस्फोरस, इट विल यूटिलाइज दैट फॉस्फोरस इन बिल्डिंग इट्स ओन डीएनए, इन मेकिंग इट्स, मतलब फॉस्फोरस जो लिया रेडियो एक्टिव वो उसके डीएनए का पार्ट हो जाएगा, बट नेचुरल है। इसके बाद इट इज अलाउड टू इन्फेक्ट द इकोलाई सेल, डीएनए एंटर करेगा इकोलाई के, along with that radioactive phosphorus will also enter the E. coli cell. फाइनली ब्लेंडिंग के बाद हमने सेंट्रीफ्यूगेशन कराया, और सेंट्रीफ्यूगल फोर्स जब लगता है तो जो डेंसर चीज़ें होती हैं दे सेटल डाउनवर्ड्स, एंड जो हल्की चीज़ें होती हैं, लाइटर चीज़ें, वो ऊपर रह जाती हैं टेस्ट ट्यूब के। तो जो जिसकी ज़्यादा डेंसिटी होगी वो नीचे आ जाएगा। बैक्टीरिया की डेंसिटी ज़्यादा है और फेज वायरस की डेंसिटी कम है, तो वायरस तो सुपरनेटेंट रीजन में रह गया और पैलेट रीजन में बैक्टीरिया आ गया। लेकिन हम ये देख रहे हैं कि रेडियो एक्टिविटी जो है वो पैलेट रीजन से एमिट हो रही है। इसका मतलब ये है कि वो जो रेडियो एक्टिव फॉस्फोरस हमने यहाँ पे यूज़ किया था, इट हैज़ एंटर्ड इनसाइड the E. coli cell, or radioactive phosphorus being a part of DNA, that also proves that DNA has entered inside the E. coli cell, proving that genetic material is DNA and not protein. तो हर्सले एंड चेज एक्सपेरिमेंट से, पूरा नाम है एल्फर्ड हर्सले एंड मार्था चेज, 1952 में जो परफॉर्म हुआ था, that proved that DNA is a genetic material एंड प्रोटीन इज़ नॉट अ जेनेटिक मटीरियल। जो पहले ऐसा सोचा जाता था कि हाँ प्रोटीन भी जेनेटिक मटीरियल हो सकता है, और ज़्यादातर साइंटिफिक कम्युनिटी दे फोकस्ड ऑन प्रोटीन बीइंग द जेनेटिक मटीरियल एज सच। तो ये आपने देख लिया। इसके बाद हम लोग कुछ और बातें डिस्कस करेंगे। वी आर टॉकिंग अबाउट, शुड आई रब दिस, आई एम रबिंग दिस, इरेजिंग दिस एज सच। वी विल बी टॉकिंग अबाउट features which a genetic material should have. What features should genetic material include? So let us talk one by one. First one, ठीक है, first one we are talking about what features genetic material should have. It should be able to replicate itself. That is the first point. It should be able to replicate itself and form its own copy. Second is it should be able to replicate itself and form its own copy. Second one is it should be stable structurally and chemically. It should be stable structurally as well as chemically. It should be stable. Third is it should provide the scope for slow mutation for evolution to occur. Now it should be inherited in form of Mendelian character. ये सारे पॉइंट्स हैं जो कि इम्पॉर्टेंट है. It should be able to replicate itself and so form its own replica. It should be stable structurally and chemically. It should provide the scope for slow mutation for evolution to occur. And the fourth point is it should be inherited in form of Mendelian characters. ये four points are very essential for a molecule to act as a genetic material. Now further we'll see how DNA is better genetic material than RNA, even though RNA can directly form protein, even though it acts as a catalyst then also. So for today, let us finish this lecture. Thank you and have a nice day.